In this video, we will read Unit 3, Cell, Structure and Functions. Biology is the study of living organisms. The detailed description of their form and appearance only brought out their diversity. It is the cell theory that emphasized the unity underlying this diversity of forms, that is, the cellular organization of all life forms. A description of cell structure and cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around living phenomena, that is, physiological and behavioral processes. This mystery was the requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take a physico-chemical approach and use cell-free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by the analysis of living tissues for elements and compounds. It will tell us what types of organic compounds are present in living organisms. In the next stage, one can ask the question, what are these compounds doing inside a cell, and in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes? It can also explain the abnormal processes that occur during any diseased condition. The physico-chemical approach to study and understand living organisms is called reductionist biology. The concepts and techniques of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is provided. Before starting chapter 8, let's get to know about this scientist G.N. Ramachandran. G.N. Ramachandran, an outstanding figure in the field of protein structure, was the founder of Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of proteins through the use of Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on October 8, 1922 in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as the top ranking student in the BSc Honours Physics course of the University of Madras. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications on the models of alpha helix and beta sheath structures that directed his attention to solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. Let's start with chapter 8, Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of 1. Independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Antony von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and the improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodor Schwann, a German zoologist, studied different types of animal cells and reported that cell had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that bodies of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained that cells divided and new cells are formed from pre-existing cells. Famous quote, omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory as understood today is: first postulate, which was given by Schleiden and Schwann, all living organisms are composed of cells and products of cells. Second one, which was an addition by Rudolf Virchow, all cells arise from pre-existing cells. Now let's see an overview of cell. You have earlier observed cell in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas cells that lack a membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane bound distinct structures called organelles, like the endoplasmic reticulum, ER, the Golgi complex, lysosomes, mitochondria, microbodies and vacuoles. The prokaryotic cells lack such membrane bound organelles. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on rough ER. Animal cell contain another non-membrane bound organelle called centrosome, which help in cell division. Cells differ greatly in size, shape and activities, as you can see in figure 8.1. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like or even irregular. The shape of the cell may vary with the function they perform. Figure 8.1 shows different shapes of the cell. This is the red blood cell, which are basically round and biconcave. White blood cell, amoeboid shape. Columnar epithelial cell, which are long and narrow. This is a trachea which is elongated, has pits on its walls. These are mesophyll cells, round and oval, and you can see chloroplast inside them. This is a nerve cell, very large, branched. This is the cell body, this is the axon, and all these branches coming out from the cell body are basically dendrites. Let's read about prokaryotic cells now. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells, as you can see in figure 8.2. Let's look at prokaryotic cell. The prokaryotic cell are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells, as can be seen in figure 8.2. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, which are rod-like, coccus, which are spherical, vibrio, which are comma-shaped, and spirillum, spiral. The organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semi-fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelle, like the ones in eukaryotes, are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of the inclusions. A specialized differentiated form of the plasma membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of cell membrane. As you can see in this figure, this shows comparison of a eukaryotic cell with other organisms. This is a eukaryotic cell. Size ranges from 10 to 20 micrometer. It has well-defined nucleus, mitochondria, all the cell organelles, Golgi body, all the cell organelles are present. And then you have a prokaryotic cell. Size range is 1 to 2 micrometer. This is the diagram of a bacteria. You can see flagella coming out of the glycocalyx. Then PPLO, pleuropneumonia-like organism, about 0.1 micrometer in size. And then viruses. You cannot see it with microscope, light microscope, 0.02 to 0.2 micrometer in size. So you can see it only with electron microscope. Now let's see cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound 3-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, viz. Those that take up Gram stain are Gram positive and the others that do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cells. These extensions are in the form of vesicles, tubules and lamellae. They help in cell wall formation, DNA replication and distribution to daughter cells. They also help in respiration, secretion processes, to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made up of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cells. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. These are not bound by any membrane system and lie free in the cytoplasm. For example, phosphate granules, cyanophycean granules and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Let's study eukaryotic cells. The eukaryotes include all the protists, plants, animals and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different, as the former possess cell walls, plastids and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells, as you can see in figure 8.3. So this is the figure 8.3 that shows a plant cell and an animal cell. As we can see, there is a very prominent cell wall present in plant cell. Cell wall is absent in case of animal cell. One more feature you can clearly see, vacuole, presence of vacuole in plant cell, but it is absent, or very small vacuoles present in animal cells, and that too present at the periphery. And vacuole here is large and is present in the center. It is so prominent that it displaces nucleus to the side. One more thing is the presence of plastid in case of plant cell, which is absent in case of animals. Plastid, chloroplast, leucoplast and all those. There is one more feature that differs between these two. Centriole is present in case of animal cell and it is absent in case of plant cell. So these are the eukaryotic cells. You can see that prominent nucleus is present in both. Double layered nuclear membrane is present around the nucleus. All the cell organelles, peroxisome, lysosome, ribosome, mitochondria, rough ER, and then smooth ER are present in both the cells. Let's now look at individual cell organelles to understand their structure and functions. First, cell membrane. The detailed structure of the membrane was studied only after the advent of electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment, as can be seen in figure 8.4. In addition to phospholipids, membrane also contain cholesterol. This is the figure 8.4 fluid mosaic model of plasma membrane. You can see bilayer of lipid. This is the inner layer, this is the outer layer, and this is the hydrophobic tail which is embedded inside, and this is the hydrophilic polar head. Now, proteins are embedded in the sea of lipids, all kinds of protein. This is integral one which is crossing the membrane clearly, right? These are the extrinsic ones which are present only on the outside of the membrane, right? Peripheral proteins here. And some sugars are attached to these peripheral protein, these yellow structures that you are seeing. So this is the fluid mosaic model of plasma membrane. Mosaic because proteins are not in a regular pattern, they are embedded, mosaic pattern is there. Fluid because it's not a static structure, it's fluid, it's dynamic. Lipids can move in, this lipid can flip and come inside, this lipid can go laterally also here and there. So plasma membrane is a dynamic structure, that's why it's fluid mosaic model of plasma membrane. So let's go back. Later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model, as can be seen in figure 8.4, and we have just seen that. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. So proteins can move within the membrane, lipids can also move. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. For example, sodium potassium pump. Now cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, ER, Golgi complex, lysosomes and vacuoles. Since the function of the mitochondria, chloroplast and peroxisomes are not coordinated within the above components, these are not considered as part of the endomembrane system. The ER, endoplasmic reticulum. Electron microscopic studies of eukaryotic cell reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm, that is called the endoplasmic reticulum, ER, as can be seen in figure 8.5. See here, this is the network. This is rough ER because ribosomes are present on its surface, right? This is the nuclear pore, nucleus, nuclear membrane, and then this is the nuclear pore. This is nucleoplasm, and this is basically rough endoplasmic, a network like structure. And as you go away from nucleus, you will see smooth endoplasmic reticulum which does not have ribosome on its surface. Let's go back. Hence, ER divides the intracellular space into two distinct compartments, that is luminal inside ER and extraluminal cytoplasm compartments. This is luminal inside ER and then rest is cytoplasm outside, extraluminal. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum, RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum, SER. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth ER is the major site for the synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER. Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer diameter. You can see this figure 8.6, and these are the sacs, cisternae, which are basically arranged in form of a stack. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming side face and concave trans or the maturing phase. The cis and trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans surface. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Lysosomes. These are membrane bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases and carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. Vacuoles. The vacuole is the membrane bound space found in the cytoplasm. It contains water, sap, excretory product and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of number of ions and other materials against concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer and length 1.0 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. That is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called the cristae, singular crista, towards the matrix, as can be seen in figure 8.7. You can see in this figure, this is the folding of the inner membrane and it is forming a structure called crista. An entire inner membrane is folded like this. So this is the matrix in the mitochondria. This is the inner membrane folded to form cristae. This is the outer membrane and this is the space between two membrane is the intermembrane space. The cristae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouses of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosomes, 70S ribosomes and the components required for the synthesis of proteins. The mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast and leucoplast. The chloroplasts contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoids pigments like carotene, xanthophylls and others are present. This gives the part of the plant a yellow, orange or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with the stored nutrients. Amyloplasts store carbohydrates, that is starch, for example potato. Elaioplasts store oils and fats, whereas the eleuroplasts store proteins. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid or even ribbon-like organelles having variable length 5 to 10 micrometer in length and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, which is an alga, green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma, as can be seen in figure 8.8. So this is the structural view of chloroplast, outer membrane and then the inner membrane. And these are these membranous sacs stacked one over the other. This is one sac is thylakoid and the stacks of thylakoid is called granum. Inside the chloroplast, the fluid that is filled is stroma. Okay, one more thing is these stacks of grana are attached by lamellae. You can see this lamellae. This tubular structure here is called stroma lamellae. Now, thylakoids are arranged in stacks like the piles of coins called grana, singular granum or intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, 70S, than the cytoplasmic ribosomes, which are 80S. Ribosomes. Ribosomes are granular structures first observed under the electron microscope as dense particles by George Palade in 1953. They are composed of ribonucleic acid, RNA and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunit, as can be seen in figure 8.9. Here you can see this is the smaller subunit and this is the larger one. Separated here, larger subunit, smaller subunit assembled to form the complete ribosome. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S, Swedberg's unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia, singular cilium, and flagella, singular flagellum, are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscopic study of a cilium or flagellum show that they are covered with plasma membrane. Their core, called the exoneme, possesses a number of microtubules running parallel to the long axis. The exoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exonemal microtubules is referred to as the 9+2 array, as can be seen in figure 8.10 figure. So this figure here shows the section of cilia and flagella. So this figure here 8.10 shows the section of cilia or flagella. It shows the different parts of cilia and flagella. B shows the diagrammatic representation. As you can see, this is the central microtubule, complete one. This is the peripheral tubule. One is complete, one is incomplete. This is the plasma membrane. Peripheral microtubules are in form of doublets and they are connected to each other also by interdoublet bridge. The central microtubules are the complete ones. This is the center sheath, and all peripheral microtubules are connected to this central sheath by radial spoke, a proteinaceous structure. The central tubules are connected by bridges and is also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from the centriole-like structure called the basal bodies. Centrosome and centrioles. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of peripheral triplets by radial spokes made up of proteins. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during the cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus, stained by the basic dyes, was given the name chromatin by Fleming. The interphase nucleus, that is the nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, singular nucleolus, as can be seen in figure 8.11. This is the figure 8.11 showing the structure of nucleus. This is the nuclear membrane with pores present in between. This is not a complete full membrane. It has interruptions in between because pores are present. This is the nuclear pore. This is the nucleus, membraneless structure present in the nucleoplasm. It has ribosomal RNA, some amount of protein. It is basically responsible for the synthesis of rRNA. This is the nucleoplasm and this entire structure is nucleus. Electron microscopy has revealed that the nuclear envelope, which consists of 2 parallel membranes with a space between, them 10 to 50 nanometer of space, and the space is called perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of the places, the nuclear membrane is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, for example erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 46 or 23 pairs of chromosomes. You will study the detail of DNA packaging in the form of a chromosome in class 12th. Every chromosome, visible only in dividing cells, essentially has a primary constriction or the centromere on the side of which disc-shaped structures called kinetochores are present, as can be seen in figure 8.12. This is the structure of a chromosome. This is one sister chromatid and this is the another one. Two sister chromatids are basically joined by at a primary constriction called centromere, and on both sides this protein plate is present called kinetochore. This is the plate where spindle fiber will come and join. Going back, centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosome can be classified into 4 types, as can be seen in figure 8.3. The metacentric chromosomes has middle centromere. Centromere is present in the middle, forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. So this is the submetacentric, this is the shorter arm and this is the longer one. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm. So this is the acrocentric, one very short arm, other very long arm. Whereas the telocentric chromosome has a terminal centromere. This is the telocentric chromosome and the centromere is terminal. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives the appearance of a small fragment called the satellite, as can be seen here. This is the secondary constriction and this is looking like a satellite. Now the microbodies. Many membrane bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Now the summary of this chapter. All organisms are made of cells or aggregates of cells. Cells vary in their shape, size and activities or functions based on the presence or absence of a membrane bound nucleus and other organelles. Cells and hence organisms can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. The endomembrane system includes Golgi complex, lysosomes and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitates locomotion. In animal cells, centriole also forms spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activity of organelles but also plays a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. Helps in the transport of substances, synthesis of proteins, lipoproteins and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. The secretions of cells are packed in them and transported from the cell. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate, that is ATP. They are bound by double membrane. The outer membrane is smooth and inner one folds into several cristae. Plastids are pigment containing organelles found in plant cells only. In plant cell, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reactions, stroma of dark reactions. The green colored plastids are chloroplasts which contain chlorophyll, whereas the other colored plastids are chromoplasts which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, cell is the structural and functional unit of life.